So, what is the role of cell handedness or chirality in um, cancer metastasis? Cancer is the uncontrollable division of cells, and it becomes very dangerous when these cells begin to metastasize or travel to different parts of the body via the bloodstream. Cancer may affect different parts of the body, mainly including or most common being the lungs, breast tissue, and the colorectal system. In 2020, alongside the COVID-19 pandemic, cancer alone claimed the lives of almost 10 million individuals worldwide. A mechanism known as apoptosis regulates a cell's natural lifespan. However, cancer cells lack this ability. Cellular chirality defines the communal tendency of cells to rotate in a specific direction. All cells tend to exhibit one of three states: a left, right, or neutral bias. Chirality is a relatively novel study in the field of cellular biology and bioengineering. Dr. Fahn's group, one of his earlier studies showed that a loss of chirality in endothelial cells, or the cells that line the blood vessels, will cause the blood vessels to become very leaky. Cell-cell junctions play a pivotal role in providing structure and proper cellular functions within a cell group. When cancer cells metastasize, they invade through the blood vessels, cell-cell junctions, and propagate into secondary sites. The question we sought to answer throughout our research was, how does cancer affect the chirality of endothelial cells? The hypothesis that we tested was that the physical introduction of cancerous AT1 or HER2 cells on a micropattern seeded culture of HUVEC cells or human umbilical vein endothelial cells will disrupt their inherent chirality. Experimental design. We started with a gold glass slide with a polymer stamp to stamp on micropatterns onto these slides using a process of involving surface adhesion molecules and blockers working in tandem to only allow cell adhesion to the parts of the slide outlined by the stamp. Our layout is as follows. We had endothelial cells, which were the HUVEC cells, the human umbilical vein endothelial cells. We had our test group, or our tumor cells, which were 10A, AT1, and HER2. Both AT1 and HER2 cells are derived from the 10A cells, with AT1 having HRAS and HER2 having ERBB2 overexpression genes. We tested these cells into two groups: a non-contact and physical contact group. In the image on the left here, we see the non-contact group using a Maxwell filter to separate the tumor cells from the regular cells, only allowing signaling pathways to be exchanged between the two and not actually physically touching. The tumor cell concentration was 10, 50, and 100,000. So here is kind of the method that our MATLAB program used, our coding program used to define chirality. So using the circumferential angle with the cell, if we found that the angle was positive, it would be noted as counterclockwise. If it was a negative angle, it would be noted as clockwise, and anything in between would be non-chiral. Our results. After a process of cellular staining, imaging, and graphical analysis after the cells have reached a great enough confluency using MATLAB and ImageJ, it was found that for the physical contact groups, as the concentration of cancerous cells increased, the HUVEC cells' chirality gradually became weaker or partially reversed. The cytokine groups showed little change. So here are our results. So the 10A group with the endothelial cells, non-contact and physical contact, we see that there was little change in the non-chiral and counterclockwise biases. As we go on to the actual cancer cells, we see a larger increase, especially in the physical contact for the AT1, and it remains true for the HER2. We see a large increase in non-chiral and counterclockwise biases as the concentrations of the tumor cells increase. So what does this mean? Now that we know that the chirality of HUVEC cells are heavily affected by physical contact with cancer cells, further research must be conducted to determine what exact mechanism or pathway causes this weakening of chirality. The discovery of such a mechanism may prove useful in lowering or preventing the chance for metastasis. I'd like to thank Dr. Ji Fahn for allowing me the opportunity to conduct this research over the summer. I would like to thank Hanan Mohammed, my research partner, big help in making sure the cells are healthy and just overall big help. Thank you, Cher, for allowing me to this opportunity to conduct research over the summer. And thank you all for listening.